Good evening, everyone. Today is your week 5 session. So please let me know if there is any issue with the screen sharing or my voice or the network. So in week 5, you have covered few topics related to machine learning and clustering. You have also looked into the hypothesis testing. You have also looked how to use ProTIGY and what are the purpose of this ProTIGY. And then you have also looked into the study of proteogenomics, in which we can identify different proteoforms and what is the main purpose of looking into the proteoforms. So in this session we will be practicing few MCQs in the end. We will look into the current literature which is available related to different machine learning techniques, and I'll take your doubts if you have any regarding your assignment or any of the topics which will be covered in this session. So firstly I'll start with the machine learning. So when we talk about machine learning, before moving to machine learning, we must be aware of the main topic, which is artificial intelligence, which is a broader term. So artificial intelligence, it involves multiple techniques which try to replicate the human behavior. So how humans respond to different problems, how humans think, how they take decisions. So artificial intelligence is like how human intelligence is there. So in that way we try to mimic that. So that is known as artificial intelligence. And under artificial intelligence there are different terms or different techniques which comes. So machine learning is one such technique in which we try to integrate that artificial intelligence into the machines. So when we devise some techniques to identify certain hidden patterns in the data and predict certain things, so that is known as machine learning, where machine is learning new things and it is taking some decisions based on the previous data which we already have. Then comes deep learning. So deep learning is a smaller term which comes under machine learning. So we call deep learning when we talk about the neural networks. So they are specific type of machine learning technique in which behaves like a human neurons. So as we see in human neuron, we have dendrites and axons. So how the signals goes from one neuron to the other neuron. So I hope you are aware about the sensory organs. So how they respond. So whenever you means touches a hot pan, then you take your hand back. So that is, you're touching something, that signal is going to your brain which is telling you that which decision you should take. So that kind of signaling where one information is passed on to another cell, then it is continuously passing through layers. So that is known as deep learning, when multiple such neurons are attached. So neural network comes into play in deep learning. Then comes generative AI. So I hope till now you have seen chat gpt and different other search engines, so other than Google and other things. So generative AI is a smaller term under deep learning in which it tries to generate new text and it tries to capture the information from different sources and brings a shorter abstract form to you so that you can get the broader picture in a shorter words. So you don't have to search through multiple pages. It simply takes information from multiple sources and gives you a summarized picture. So that is generative AI, and not just text, but it can also gives you images. For example, nowadays you can also create any image artificially. So all such things comes under generative AI, where something is generated with the help of artificial intelligence. So these are few terms, and there are other terms also which are coming day by day with the advancement. So now one thing which you must know, that how this machine learning works. So just I'll check. Okay. So in machine learning we have a training data because which is a prerequisite for machine learning. So we must have some data. So in this case we have a training data. We have to train machine learning algorithms. So what machine learning algorithm does, they study the data, they analyze the data, and they tries to identify that what is the relationship between that data. So in that process machine learning algorithms are trained, and then they model the input data. So on the basis of data which we have, they try to design a model such that if any new information comes in, then it is able to identify that information. So model the input data, and then machine learning algorithm is devised. So algorithm is a kind of a procedure, stepwise procedure, which tells that what is the relationship between your input data set, so that whenever any new input data set comes, it will apply that equation or apply that function to that new data and will characterize, or it can predict. So later on we'll look into different types of machine learning. At that time you will be more clearer that what does machine learning algorithms mean. So new input data will come, and then machine learning algorithms which are based on the training data, they will apply certain equations. They will try to figure out that what is this new input data. So for example, let us consider that in training data we have photographs of multiple fruits. So when we will be training machine learning algorithms, so then we will be giving some characteristics, that what is the characteristics of particular fruit. And for example, we have a labeled data in which a fruit image is given, and we also know what is that fruit. So in such scenario, we will be associating certain terms with a particular image. So whenever you give a new data, which means any new picture which might have certain fruits, so on the basis of previous information, it will try to predict that which is that fruit. So in this case, machine learning algorithms will try to predict that input data. So now what we do is we check for the accuracy, that whether the prediction is accurate. So if it is accurate, then it is a successful model. But if it is inaccurate, for example, you are giving the image of an apple and it's predicting to be orange, so that means there is something means problematic with your model. You might not be capturing properly, so you have to again start with the training of machine learning algorithm. So we will see that how this training is done, how different features are there which decides that how the input is related to a certain output. So we will see that one by one. So I hope till now everything is clear. Okay, thank you. So now the another thing which we must understand is that why we are using this machine learning in proteomics, because till now you might have seen multiple examples of machine learning with respect to day-to-day life uses, but in proteomics, why do we use it? So we can use it for the protein identification and its quantification. So we can also check that if we have a sample, so which are the proteins which are present in the sample and in what quantities. So this quantification and identification is in respect of mass spectrometry. So in mass spectrometry, when peptides are taken, so how those peptides are related to certain proteins, and how number of peptides will decide the abundance of a protein. So all such things can also be identified with the help of machine learning algorithms, because it will be smoother process. You don't have to do it manually one by one. Or so in machine learning, you can give multiple samples at the same time, and it will predict the quantities as well as the proteins which are present in the sample. So it will identify. And then second thing which you can do is that you can also improve the accuracy of protein identification from mass spectrometry data. So if you have a huge amount of data, then you can see that what are the issues. So in machine learning model, you can try to resolve such issues. Then comes protein structure and function prediction. So there is a app known as AlphaFold. So it has been based on the deep learning. So in that, what it does is it takes the amino acid sequences and it tries to predict its three-dimensional structure. So earlier time, what used to happen is that students used to study the protein structure, and it used to take a lot of time for experimental identification of the structure, such as X-ray crystallography or NMR. So with the help of this machine learning and with a lot of data which is previously available, so because data is a must, if you don't have data, you cannot use machine learning. So one thing is that we already had a lot of data of protein structures. So what machine learning does is that it compares the sequences and the structures, and it tries to identify that which sequences are most likely to generate what kind of structure. So with that, AlphaFold is completely based on multiple sequence alignments and how that sequences are giving rise to similar type of structures. So in that way, it is able to predict the structure of protein. So protein structure prediction can also be done with the help of machine learning. And then if we talk about function, it is not that much into use, but yeah, to some extent we can predict the function of uncharacterized protein, because what is believed in the proteomics, that the proteins which have nearly similar structure, they might have nearly similar sequence, they might have similar structures, and the two proteins which have similar structures, they might perform similar functions, not identical but similar functions. So to some extent we can predict what could be the function. Then comes protein-protein interactions. So protein-protein interactions can also be predicted with the help of machine learning. We can identify that what are the characteristics of two proteins to interact. So in that way, it can help us to predict more and more protein-protein interactions, and it can also help us to predict the potential protein interaction partners for a given protein. So these are all based on the comparisons and classifications. Then genomic variants impact on the proteome. For example, you already have different variations such as SNPs or missense mutations, and you know that these are involved in this particular disease, and you also have some kind of idea that how they affect what kind of pathways they alter. So in that way, if you have that information with you, so whenever a new protein comes in and you already have the idea of the mutations or changes which are present in that protein, So to some extent you can also predict its effect. So that things can also be facilitated by machine learning, and you can also predict novel protein variants that arises due to mutations in the genome. Then if we talk about multiomics integration, so proteogenomics is one among them, multiomics. So we can also integrate proteomics and genomics to better understand a disease mechanism, because we, as in previous all sessions we have seen that proteome is quite dynamic. So by only looking at proteome we cannot get that much information, we cannot rely on that. And if we look at the genome information, then it is quite static, and in different tissues different proteins are expressed, so we cannot rely on any one of them, so we have to combine them so that we can get better conclusions. So in that way multiomics integration can also be facilitated by machine learning, and it can also help us to identify key regulatory mechanisms which generally link genome, transcriptome and proteome. So this is all about the use of machine learning in proteomics, and I know there are many other uses as well, but these are the key uses which I have highlighted in this slide. So now we will look into different types of machine learning. So there are other types as well, but we will be focusing on supervised and unsupervised for now. So supervised learning is, as the name suggests, it is supervised, which means somebody is supervising it. So it is generally task driven. When you have some task, you have to predict something, or you have a labeled data. For example, I have a set of data which is already labeled, and whenever a new data will come, so I have to predict its label. So that thing is a supervised learning. Unsupervised means you do not have any information, you simply have a data. Now by looking at the data you have to identify certain patterns, so that thing is known as unsupervised learning, when you don't have any labels, but what you try to do is you try to identify you try to cluster it. So you try to identify which of the data is clustering together, so that you can divide it into multiple clusters. So that comes under unsupervised learning. And then comes reinforcement learning. So there is another type of learning which is semi supervised, so in which a little bit of information is already known, and so we won't go into that detail. Then comes reinforcement learning. So reinforcement learning is, it is like a trial and error basis learning, in which you learn from the mistakes. So whenever a model will make some prediction, if there is some error, then we will introduce the penalty. So these learning techniques, what they ultimately do is that they design an equation. So in that equation, whenever a new value will come in, it will apply that equation and tries to give you output. So if your output is not right, if you are checking for the accuracy, then what you will do is, in that model you will add some penalty. So by looking at that penalty and rewards, it will try to means improve the accuracy of the model. So that is about the reinforcement learning, in which there are treats or rewards as well as penalties, from which the model again and again refines itself till the point the accuracy has improved. So we will be focusing on supervised and unsupervised for now. So firstly let's talk about unsupervised learning. So before moving to it, I want to ask that whether till now everything is clear. Okay, thank you. Yeah, so now when you will be going through unsupervised and supervised learning, you will, at the end of this session you will be cleared with all your doubts related to machine learning. So when we say unsupervised, first thing which you must keep in mind, that it is not supervised. That means the data which you will be using is unlabeled data sets. When we say unlabeled, what does it mean? For example, in the figure if you see, this is a training set and we have multiple fruits in the training set. So in this case I'm giving example of fruits for simplicity. You can also consider proteins as an example. So in training set we have multiple fruits, but we do not have labels. Labels means if it would be a supervised data set, then there would be labels such as this apple would be labeled as apple, banana would be labeled as banana, but in this case you simply have these figures, you do not have any other information that what does it mean, and this training set don't tell you that this is apple or this is banana. So now in unsupervised learning, the task is, it has to identify, it has to cluster similar looking objects, it has to identify that which objects are different and which are similar. So it tries to discover hidden patterns and data groupings without the need for the human intervention. So in this case this is a training set. So now how to identify or how to cluster? So what it does is it uses the feature vectors. So feature vectors in this case could be the color. So for example, in the training set, what information you will be having is, you will be having the color of each of them. So by color you can distinguish between them, or size or shape could be a feature. These are just for example, there could be other features as well, or shape, weight, texture and sweetness. So these are some kind of features with which you can distinguish among themselves. So then you will use the machine learning algorithm, you will design that algorithm is simply a stepwise procedure which tells you that okay, so if I want to identify apple, so it can be either green or it can be either red, and its size, so there will be some centimeters of this particular centimeter belongs to apple. For example, you can see that watermelon is large and apple is smaller compared to watermelon. So in that way each fruit will have a different size, it will have a different shape, its weight might vary, its texture might vary, its sweetness level might vary. So by looking at these characteristics, the machine learning algorithm will decide that okay, if this is the color, this is the size, so the size won't be fixed, it could be in some range, so it will decide, and then the predictive model will be developed. So in the test set is a set in which you give the input, and now our model will try to predict for that test set. Training set is the one in which you train your model, in which you have the information such as color, size, shape, every feature you already have, you try to figure out that what is the relationship of these input variables, and then in the test set you are given with certain objects or certain figures, and you try to predict what could be this fruit, in which category it will fall. So it will cluster the test set, so these will be the three main clusters. So it is trying to cluster it into three clusters here. So when we will look into the clustering algorithms, then also we will understand that why here are three clusters, because it could also have five clusters. So it depends on you that how many number of clusters you want to see. So in this case the cluster size was 3, so that's why it clustered these things into 3. Now let's see the next. So in unsupervised learning there are two main techniques, one is cluster analysis and another is dimensionality reduction or dimensionality reduce. So in cluster analysis, as I have talked earlier, that in unsupervised learning you try to cluster it because you do not have labels, so you cannot predict anything, but you can cluster them by looking at already available data set. So for clustering you can use k-means clustering, hierarchical clustering, Gaussian mixture model or density based clustering. Whereas in dimensionality reduction, principal component analysis, independent component analysis and factor analysis, and I hope you have gone through the principal component analysis in the week 5. So we will little bit look into other as well. So now let's look into cluster analysis. So when we talk about clustering techniques, so as we have seen there are multiple clustering techniques. When we say k-mean clustering, so as the name suggests, k means, so k stands for number of clusters. So in this you have to decide how many clusters you want to see in the data set. So you have some input data and you decide on the basis of those number of clusters, it will try to cluster your data. So how clustering is done, it will try to see which features are more closely resembling to one another, so it will be taking the means. So by looking at those means, it will cluster your bigger data into smaller clusters. So that is about k-mean clustering. And means how you can use it in proteomics, so you can use it to group the protein expression profiles from different samples. And advantage of k-mean clustering is that it is simple, it is efficient and it is scalable, so you can use it on a huge amount of data, it will not create any issue. But the limitation is that you have to specify the size of the cluster, which is k, because in some cases what you have is you have a lot of data and you don't know how many clusters could be present. As we have seen in the previous case, it was so when it was clustering, it has clustered orange and lemon to a same single cluster. So now you are not aware how many clusters will be present. For example, you want to segregate them, you want to see them in different clusters because they are different fruits, they are not same fruit, although they both are citrus fruits, but they are not same fruits. So in that case, in k-mean clustering, it's quite difficult to identify that how many potential clusters would be present. So in this you have to specify k value, so which could be a limitation. Then comes hierarchical clustering. So hierarchical is a concept which is like when you are following some kind of hierarchy or some kind of tree, that it has some roots, then it has branches, leaves. So in that way, whenever clustering is done in such perspective, that is known as hierarchical clustering, and it is also divided into two categories. So in this case, it tries to design or build a tree kind of a structure, which is also known as dendrogram. So it can either merge or split the clusters. So if two clusters are nearly similar, it will merge them into a single cluster. So in the later slides we will see that how it does it. So whenever two smaller clusters are merged to a bigger cluster, it is known as agglomerative hierarchical clustering, because it is trying to agglomerate, means combine, aggregate. Then comes splitting or divisive clustering. So in this case it will try to devise, means if you have a bigger cluster, it will again split it into smaller clusters. For example, as the previous example, we have both orange and lemon in a one cluster. So what divisive clustering will do, it will cluster it into two different clusters, calling them as lemon and oranges. And if we talk about agglomerative, then it will combine them and make a bigger cluster, which will be for the citrus fruits. So that is about the hierarchical clustering. It tries to identify the relationships among proteins across different conditions or time points. So in that way we can use it in such scenarios. And the advantage of this hierarchical clustering is that, as we have seen in k-mean clustering, we have to specify number of clusters, but in hierarchical clustering you do not have to specify number of clusters. So due to that it is quite efficient as compared to k-mean clustering, but the limitation is that it is computationally expensive, because you have not specified number of clusters. So it will try to identify what could be the potential number of clusters. So in that way it will take longer time, and it is computationally expensive. That means it will means uh, consume greater amount of RAM of your system. It is not scalable. If you have a huge amount of samples, then it will create some trouble. And it is also sensitive to noise. For example, you have certain outliers, then also it will try to adjust it, into clusters. So in that case it is quite sensitive. Agglomerative clustering, as it was in your assignment, so I have kept it into a single category, but it comes under hierarchical clustering only. So it is a type of hierarchical clustering that starts with individual points as clusters and merge them based on the distance criteria. So in the example we will see later on that how it is trying to merge a smaller clusters into bigger clusters. So it is used to cluster proteins with similar expression patterns from mass spectrometry data. So in that way agglomerative clustering could be used in such scenarios, and there is no need for initial seed. So initial seed is like, you are not deciding that how many clusters you want to merge or how many clusters you want to see in the end, or from where the agglomerative clustering should start. So there is no such need. But as we have seen that hierarchical clustering has certain limitations, so similar limitations are with the agglomerative as well, because it is also under hierarchical clustering. Then comes density based clustering, and the name is the DB scan, which stands for density based spatial clustering of applications with noise. So in this, what it tries to do is that, in simple terms, as it is based on density, so whenever the region where the number of points or number of inputs are dense, where the features are closely similar, so it will try to make a single cluster of that dense region. So that's why it is based on density. So where density is high, it will make a one cluster, and wherever density is low, or the points which are far away from that density rich regions, they will be termed as noise, where the cluster size is very small, where the neighbors are not that closely present. So that is known as DB scan. It tries to group points that are closely packed and mark those points that are far from any cluster as outliers. So in this way they are able to identify outliers as well, because in hierarchical clustering we have seen that if there are some outliers, then also it will try to fit it into a cluster, it will try to keep it together, which might have some influence. But in DB scan it will term it as a noise, and it will ignore it, it will not put it into any cluster, it will simply say it is a noise, it cannot be clustered. So in this case, detecting protein groups with similar expression in noisy proteomic data. So in that case we can use DB scan. It can find arbitrarily shaped clusters and identify outliers, but the problem is that there is difficult to choose the parameters, that how to decide. So that is quite difficult, because in the later slide you will see that how DB scan decides that when to call something as a cluster, what should be the neighborhood distance. So all those things are quite difficult to decide, that which distance should be appropriate to call two points to be neighbors and group them into a one cluster. So that thing, that parameter deciding, is little bit difficult, because densities might vary. Then comes Gaussian mixture models. So it models the data as a mixture of several Gaussian distributions, so which allow to cluster to take the elliptical shapes. So as we have seen in k-mean clustering, it will try to make circular or spherical clusters, due to which it might miss out certain data points. But Gaussian mixture model is an improvement. It takes multiple Gaussian distributions and make the elliptical models. So in this case, you do not have to means fix to a circular structure and miss some data points. It will take shape accordingly. So it can take elliptical shape so that multiple data points can come into a single cluster. That is the biggest advantage of Gaussian mixture models. And in this you can use it in identifying the protein subtypes or complex expression patterns in proteomic datasets. It can handle complex cluster shapes. So elliptical is just an example. So it can handle complex shapes of clusters. It has not to be only spherical or round. And it is also known as soft clustering, whereas k-mean is known as hard clustering because it is hard, it has fixed that it should be spherical. But this is soft clustering because in this any shape can be achieved. It assumes Gaussian distribution and sensitive to initialization. So whenever if the data in your distribution is not normal or it is not in Gaussian distribution, then there will be problem. So you can only use Gaussian mixture models when your data set, when the distribution of the sample variables or data set in your sample are in normal distribution. So this is all about these techniques. Now one by one we will see each technique so that it will be more clear with examples. Yeah, so before moving forward, I'll just quickly ask. Okay, so you're asking about noise. Yeah, so in the later on examples we will be more clear about noise, but till now the noise is a outlier. So for outliers only we use another term as noise. So there are certain proteins whose expression or means there are certain peaks in mass spectra which might not be the peak coming from the protein, which might be a contaminant peak, or there are certain proteins which are means overexpressed in some sample which are not for means which you cannot cluster into the already available other proteins. So in that case those are termed to be noise, means which are not closely related with the already available data, because in unsupervised learning your aim is to cluster your bigger data to smaller clusters. But what if in your entire proteomics data set one protein is behaving entirely different, its expression is very high or its expression is very low as compared to other proteins which are present, so you cannot cluster it into those already available proteins. So in such scenario that protein will be taken as a noise, you will keep it separate, you cannot cluster it, because a single protein cannot, a single data point cannot make a cluster. So you have certain thresholds that if 3 data points are present or if 4 data points are present, then only you will call it a cluster. So in such scenarios the noise is where you do not have enough number of data points to call it as a cluster, and it behaves very differently from already available data set, so you will term it as a noise. Okay, so now let's see what is k-mean clustering. So in k-mean clustering you will try to cluster your data set, but you have to mention that what should be the size of the clusters, how many clusters you want to make. So there are 5 main steps in k-mean clustering. Firstly you have to decide or specify number of clusters, which is known as k. Then you have to randomly initialize the cluster centers. So for centering, because as we have seen mean, so mean is used for centering your data. So when you have a huge amount of data you will randomly decide some center point. So for that what you do is you will look at the distribution of your data set, and then you will try to figure out where the multiple data points are present and what should be the center. So this assignment is random, randomly you decide some point to be a center, so that from there you can calculate distance of other points. So this is kind of assigning randomly. So firstly random assignment will be done. So for example here they have thought of 3 clusters, this is randomly assigned, some midpoints are selected which are mu1 mu2 mu3. So previously they are not mu1 mu2 mu3, because mu1 mu2 mu3 are generally used to represent the means if you are looking at some sample distribution. So these are the so this particular variable with that is mu is used to show or used to depict the mean. So firstly you will take the center points randomly, then what you will do, you will calculate its distance to all the data points, and you will so as we can see in the step 3, assign each data point to the closest cluster center. So as you have decided center points, now you will calculate distance of all other data points which are present, and you will see that what is the distance of those data points from the center which you have decided. Now you will recompute the cluster center as the mean of all the data in that cluster. So after deciding that center point, the distance from all the data points will be calculated, and then all the data points which are present in certain range to that center point, they will be kept into a single cluster. For example, in this case they have decided 3 clusters, means 3k, that is 3 clusters. They have 3 center points. Now they are checking the distance. For example, this point which is present here, it is far from the blue colored dot, whereas it is closer to red dot, so it will be in a cluster of that red dot. So similarly, on the basis of distance, all the data points are put into some cluster. Now the data points which are present in that cluster, we will take their mean. We will see that all the data points falling in one cluster, what is the mean of all those data points. If their mean comes out to be the centroid which we have decided, then it's fine. If not, then we will adjust the centroids. So in that way we try to recompute cluster centers in such a way that the distance from that center, so that should lie in the center point. So the centroid should be the center point of my entire cluster distribution. So in that way, that is one thing which has to be make sure in k-mean clustering, that the centroid should be a center point from all the data points that are present in the cluster. You will repeat these 3 and 4 step till the time the means you do not see any difference. Because if your mean is different and your centroid is different, then you will keep on changing the center point, so that when you calculate the mean of all the data points in that cluster, it should be same. Another limitation of k-mean clustering is also that in the k-mean clustering you also make sure that the number of data points which are present in each cluster, they should be uniform. You should not see that one cluster is having huge amount of data and other cluster is having very low data. So in this, it also makes sure that the circles or the cluster size, it should be uniform. It is not that much variable. So by recomputing and all those things, there are certain boundaries which are defined. So if you have to divide it into 2 sets, then this will be the boundary. If it is divided 2 versus 3, so there are certain boundaries. This is all the repetitions. You are just trying to figure out what should be the proper centroid, and ultimately there could be 3 clusters which were identified. So these 3 clusters were most accurate ones, because your aim is that the distance of the points from the centroid, it should be minimum as compared to other centroids. So in this case, they were able to figure out that all these data points are lying different or lying at certain difference from other clusters, so that you are not confused. For example, if some data point is lying at a similar distance from 2 data points, so in that case it will create some trouble. So that is one thing in k-mean clustering. You try to make your model such that there should be no such confusion. Your centroids should be far apart from one another, so that you can easily distinguish these data points which are present in your distribution, due to which it is sometimes hard to achieve. Okay, then let's talk about hierarchical clustering. So in hierarchical clustering, as we have talked, that you can also go for agglomerative clustering or top down, which is known as divisive clustering. So now let's talk about bottom up or agglomerative clustering first. So in the left side, as you have seen, these are few data points. These are total 7 data points starting from P0. So firstly, what it does is it sees that which data points are closer to one another. So when I say closer, what in reality it means, there are some features associated with the input data. So if features are similar, those data points will be kept in one cluster. So on the basis of that, if you look at this distribution, it means the points which are lying next to one another, they share some kind of similar features. So in this case, 5 and 6 are closer to one another. So 5, 6 can be in a one cluster. If we talk about 5, if we calculate the distance of 5, 6 from the other data points, then they are closer to P4. So then P4 will come. Now I am talking about agglomerative clustering, which means from smaller clusters I am going to merge them and make a bigger cluster of all the data points. So now 4 is closer to 5, 6, so they all will get into one cluster. Now if we calculate their distance from other data points, then they are closer to P3. So now P3, P4, P5, P6, they will come into one single cluster. Now let's look at P0. So P0 is lying quite away from everything, so it will be an entire single cluster. Then P1 and P2, they are closer as compared to other data points, they are lying closer to one another, so they will be a single cluster. Then if we calculate their distance from P0, then these both are closer to P0 as compared to other data points, so they will get into a single cluster, so we can also merge it. And ultimately P0, P1, P2, they and P4, P6, P5, P3, they can also be combined into one single cluster. So in the end we have all the data points in one single cluster. This is known as agglomerative clustering, when you are trying to merge all the smaller clusters into a bigger cluster. Whereas if we talk about top down, then what we do is we have all these data points, we are trying to divide it or split it into smaller clusters on the basis of the distance or the features which they share. So similarly, instead of moving bottom up, we will be moving top down. So why it is bottom up? Because in bottom we have multiple clusters listed, and as we move up we try to merge them. That's why it is known as agglomerative and bottoms up. When we say top down, which means from the top we have a bigger cluster and we are splitting it into smaller clusters as we move down. So in that way you can remember it, that on the x-axis there would be multiple clusters, so it will be referred to as down, and in the up there will be a bigger cluster, or we can say a data set which we are trying to cluster. So in top down it is a divisive hierarchical clustering, and bottom up is a agglomerative hierarchical clustering. So in this slide I have put a GIF to show that how agglomerative clustering happens. So as you can see, the data points which are closer to one another, they are captured into single cluster, and in the end both these data points are also kept in a single cluster. So you can go through once again these. Okay, these are closer than these are, then these both are close, that's why single cluster is formed. Now these two are close. So in this way agglomerative clustering tries to merge smaller clusters, and similar will be the case for divisive as well. Now let's talk about density based spatial clustering. So in density based clustering you have to mention that what should be the minimum number of data points that should constitute a cluster. So in this scenario, minimum points which will constitute a cluster is decided to be 3. So it is decided by the user, so on the basis of which it might alter number of clusters which are formed. So now what it does is it will consider a data point as a center, and now it will make circles around that data point, so that circles will have some radius that will also be decided by the user, which is also known as EPS, epsilon. So epsilon is the radius of that particular circle. So now what it will do is around each data point it will make a circle. As you can see all these black circles, so they are having radius of epsilon, and epsilon is decided by the user, it is the distance that specifies the neighborhood. So now how it make clusters, it will make circles around each data point, and whenever any of the other data points circle will overlap with this given circle, then we will call it to be the neighbor. So in that way all those data points which will overlap with my circle, I will make them into a single cluster. So similarly all these data points, their circles are quite overlapping, so in that way they all are getting into a single cluster, which is cluster 1. Similarly on the right side also, these all data points, their circles are also overlapping, so they are also constituting cluster 2. Whereas if we talk about these green data points, then these are the boundary molecules, because in this case we have decided that our minimum point should be 3, which means that if there are 3 points in the neighborhood of my data point, then only I will call it as a cluster, otherwise I will call it as a noise or outlier. But in this green case what you see, there is a circle which is overlapping with this blue dot, which is another data point. So in this case it has 2 points in its circle, or in the epsilon, that particular distance. So in this case these are the boundary molecules, we cannot call them noise, so we are considering them to be part of the clusters. In certain scenarios, if you have stringent conditions, if you have filtering criteria to not consider any cluster below 3 data points, then they will not be considered as clusters. but in this case they are regarded as boundary molecules and they are also considered to be part of the clusters. So similarly this particular is considered to be part of cluster 1 and this green dot is considered to be the part of cluster 2. Whereas if you see these orange dots, they are the outliers because in their circle no other data point is overlapping. That's why they are only single points and minimum criteria should be 3 or 2. So that's why these are regarded to be as noise and they will not be put into any of the clusters. So this is one of the advantage of TB scan, that you can figure out your outliers and noise and you can reject it. And it can also handle numerous features when you are drawing similarities. And in this case, as in k-mean clustering, you have to figure out that how many clusters you want to see, but in this case you do not have to mention number of clusters because on the basis of epsilon or the distance it will itself decide that what should be the number of clusters. So are you following or is there any doubt? Okay. Yeah, so now let's move forward towards Gaussian mixture models. So as I have talked earlier that in Gaussian mixture models they are advanced models. So first assumption is that your distribution should be a normal distribution, and second thing is that it is merging multiple normal distributions. So in that way it is able to adopt the elliptical shapes. So in this case if this is a cluster with different variances, so what k-mean clustering will do, it will try to make single circles and it will consider these so now after having those defined circles it will only be able to make these number of clusters. So in this case there are 4 clusters, whereas if you see the values or the data points which are near to this blue, they are also not considered to be clustered under these blue dots, they are under grey dots or they cannot be clustered. So in this case there are only 3 clusters which were identified, whereas in Gaussian mixture models as it can also take the elliptical shapes, so it is better able to capture different clusters. So in this case the dots which were previously not detected in this cluster, in green cluster, which are now identified in a better way in Gaussian mixture model because it can take elliptical shape. So in such scenarios Gaussian mixture model is a correct clustering measure as compared to k-mean clustering because you do not have to mention number of clusters and it can also take elliptical shapes. So that's why you do not have to miss on certain data points which you miss in case of k-mean clustering. So this is one such example. So in this case in this GIF they have shown that how the clusters are identified in case of Gaussian mixture models. So they are taking elliptical shapes, and if so the darker dot if you see in the middle, so if you go for the k-mean clustering then you won't be able to capture the outer circles, so outer data points, whereas if you talk about Gaussian mixture model you are better able to means change the shape of your cluster in case of elliptical as well. So that's why the data points which are lying far from the center, those points can also be captured. So you do not miss any data because in k-mean clustering you are calculating the distance and then identifying centroid, so due to which many points which are far away from the center but might have involved in that cluster will not be missed in case of Gaussian mixture model, which might have missed in k-mean. So that is one of the advantage, and it assumes that there is normal distribution of the subpopulations, and mixture models in general don't require knowing which subpopulation a data point belongs to, which allow the models to learn. Yeah, so that is advantage of Gaussian mixture model. Then comes the second category of techniques which are dimensionality reduction. Okay. Yeah, so yeah, that is one of the disadvantage with Gaussian mixture models, that as they can take elliptical shapes, so in certain scenarios if your distribution is not normal, because I have talked that main assumption of your Gaussian mixture model is that your distribution is normal distribution in which there are no outliers, but if there are outliers there is noise, then which means your distribution is not normal, it is skewed. So in such scenarios Gaussian mixture model won't be working fine. So in that case it will its clustering will not be proper. So that thing you have to make sure that your data is normally distributed, otherwise Gaussian mixture model will capture the outliers as well and it will means disturb your clustering. It will influence it in bad way. So now the second category of techniques are dimensionality reduction techniques. So in this we will be mainly focusing on principal component analysis, and rest other techniques, if you want to go through them then you can go through them. So first thing is principal component analysis. So what it does is, it if you have a high dimensional data set, then it tries to reduce its dimensionality to smaller dimensions. So when we say dimensionality, it means how many x's you have in your data set. If I have to simplify it, then for example you have x axis, y axis, z axis, then in PCA you will only have x and y axis, or you can simply have just x axis. So that is all about principal component analysis. You try to reduce your data to smaller number of dimensions, because in machine learning you have multiple dimensions, or you can say you have multiple features. Because what are dimensions? They are some features on which your data points are arranged. For example, if you have height, weight of a person, color of a person, which area he belongs, so all these are dimensions. So now what you do is, in principal component analysis, you try to identify that which of the features are mostly varying. For example, if I am taking example of a single area, for example I am only taking example of Indians, then I cannot distinguish them on the basis of means of their hair or color. I cannot broadly distinguish them. But if I compare Indians with some other country like China, then I can so their eyes would be different, their hair texture would be different. So in that case those dimensions are variable, but if I am talking about only Indian, so that dimension won't vary much. So in principal component analysis I will only keep those dimensions in which data points are varying a lot. So that's why I am reducing my dimensions, so that I can better analyze my data, because those dimensions are only increasing my means amount of data. They are not helping me anyhow to identify or cluster my data. So that is one of the advantage of principal component analysis. You identify the principal components. Principal components are the directions, or they are the axes which are capturing max variation in the data points. So in the example it will be more clear when I will take you forward. So where you can use it, you can use it to reduce the dimensionality of large proteomic data sets to identify patterns or trends. So for example, if I have taken the sample from normal and a patient who is a cancer patient, and when I am comparing their proteins, so there might be certain features which do not vary at all. For example, the expression of the proteins, for example those expressions are similar, but something else is varying, some quantity is varying, or degradation is faster. So in such scenarios I have to decide that what are the features which are varying. I will only focus on those features, and I will only consider those proteins which are varying. If certain proteins are constant among normal and diseased person, I will reject those proteins, I won't consider them. So in this way I am also reducing the size of the data which is present with me for better analysis. So it simplifies your data, it preserves variance, because it is trying to figure out that there should be maximum variance among the data points which is present. So in that way it is preserving variance, and it is making your data interpretable, so that you can interpret your data, that what are the differences. And the limitation is that it is a linear method, so it may not capture the complex structures. So that is one of the limitation. Similarly, there are other techniques as well which are trying to reduce your data from high dimensions to lower number of dimensions, so that visualization of data set and its interpretation and analysis is simplified. So this is all about these methods. If you want to go through them, you can see the slide, I won't go into detail. Now let's talk about principal component analysis. So it reduces number of dimensions in large data sets to the principal components. So principal components are also the dimensions, but these are reduced dimensions. So it does this by transforming potentially correlated variables into smaller set of variables called principal components. So these so in this table I have shown that how you can use this PCA in case of proteomics. So first of all, we have a data set which is large and complex, and it has multiple features. So if we talk about proteomics, so in proteomics what data set we can have is that we have 100 samples with expression levels of 10,000 proteins, And these 100 samples, for example, I have taken 100 patients. So 100 different patients have 10,000 proteins which are expressed at certain levels. So I have that data set with me. Now how to go with its analysis? So I need PCA so that I can reduce its dimensions. So the challenge is that this data set is huge and it is hard to analyze because there are multiple number of features which are present. So for example, identifying important patterns in 10,000 proteins across samples. So in PCA process, what you try to do is you try to identify principal components where data points varies the most and you try to reduce the dimensions. So in case of proteomics example, what would be your principal component? It could be the key patterns, which will reduce your data from 10,000 proteins to 2 or 3 principal components. So principal components will be simply your axis where data points are varying a lot. So in the next slide we will see that how it captures. So in terms of visualization, we make a plot which samples using the reduced dimensions to reveal patterns or clusters. Similarly, in case of proteomics example, the samples will be plotted using 2 or 3 principal components. So it's up to you. You decide the number of principal components, and in most of the studies generally principal component is fixed to 2. First principal component will be the dimension where there is maximum variance between the data points. Principal component 2 will be the next dimension which have the variance but lesser as compared to first principal component. Similarly, subsequently, as we increase the principal component number, it means the number which are smaller, they have maximum variance. As we move forward, the variance tends to decrease. So there is no point in considering fourth principal component or fifth principal component. That's why mainly first principal component or second is enough to decide or to reduce your data set. So what will be the outcome? It will simplify the data that will retain the most important information which will be easier to analyze or interpret. And in terms of proteomics, it will focus on few key proteins that explain most of the variation in the data set. For example, in 10,000 proteins, when you are comparing their expressions, there might be 100 or 1000 proteins which are variable. So in such scenarios, you will only focus on such key proteins which are varying rather than considering all those proteins. So what is the insight from this analysis? It helps in identifying critical patterns and it will also guide further for the biomarker discovery or some other discovery association studies. Similarly, you can identify proteins which differentiate between disease stages or treatment responses. For example, those patients are given with some drug. So in such scenarios, you can compare their protein expressions which are varying. So in that way, you can figure out that how the drug dosage is impacting the expression, whether it is bringing the protein expression to the normal levels or it is overexpressing certain proteins, how the influence is happening. So all such things can be done with the help of PCA. In such cases, we will be having, in the end we will be having only the proteins which are most varying, so that the analysis will be interpretable and it will be simplified. And when we say principal component, so what does principal component means? These are the directions in the data that capture the maximum variance, representing the most important patterns in the data set. So principal components are known as directions, not just axis but directions, in which direction most variance is observed. So in this case, I have given the example. For example, your data is distributed among two dimensions, x1 and x2, and these are the data points. So maximum variance is not captured around x and y. So what you do is you will see in which direction most of the data points are varying, and the line which is drawn, you have to make sure that all the data points, they should have minimum distance from this line. So principal component 1 is this particular line, and now what we do is we will calculate the distance of each data point from this line. We will sum it up and it should be minimum. So in such a way, there will be multiple lines and ultimately only that line will be selected from which the distance of all the data point is minimum and maximum data points are present around that line. So that will be my first principal component. Similarly, after getting my first principal component, I will find for the second principal component the line from where each point is having minimum distance, but at the same time those data points should have some variance, they should vary. So similarly PC2 is also identified, which is this particular line. So now what I will do is I will rotate my data in such a way that it is represented in the form of x and y axis. So that's why this data is now transposed in such a way that it is shown as x and y axis. So now these data points are varying among themselves. So this is one of the purpose of principal component analysis. So this figure might not be that much clear, so we will see now real life example which will make you more clearer that how the principal component analysis is working. So in this, these are the samples. So firstly what it is doing is it is calculating the distance of these data points from the line. So for example in this GIF I have shown that we have two dimensions, one is height, another is weight. Now I am converting this two-dimensional data into one dimension, in which I have compared among height and weight which dimension is capturing maximum variation among data points. So in this I have observed that in the height maximum variance is present. So I am taking the line. So first of all what will happen is this line will be like slant line, and then these data points will be converged to this x-axis. So these data points are now spread out. I will calculate their distance from the my central line. The distance is minimum, so I will keep these points on this central line so that only the single dimension is captured. So in this case, which will be my weight, not height, sorry, it will be my weight. So they are varying across the weight. So in this way I was having weight and height both the axis, and after calculating that straight line I have brought it into a single axis which is my weight axis. Similarly if we talk about the height, so similarly I can do that for height as well. I can take the central line from where the distance is minimum. Why I am taking to this, because the difference of all the data points will be minimum. If I take line here, then distance of these points will be far away. So that's why the line will only be drawn where the distance is minimum from other data points. So similarly in this scenario, okay, so in this scenario central line is not mean because it is not trying to cluster it. What it is doing is the central line will be kind of a distance. So mean is calculated at, it might be calculated at the level of all the distances, but it is not the, because it can't be the mean actually. So in this scenario we take the we select some central point and then we calculate the distance from all these points and we sum up that distance, and if that distance is minimum then we will consider that particular line. So it is just sum of the distances where the sum is minimum. So in that way line is drawn. So in such case the line is drawn for the height, the variation is captured for the height, and the data points they are brought to a single axis. So I am bringing this point to this to here. So I am trying to reduce it to a single axis. So this is one another example of the GIF, that how it is stepwise doing it. Yeah, so now it is a mean point and then the line is changing, mean point is decided, and then so center, yeah, so this thing is the mean, but the line where you will, in which direction that line will be, that will be decided on the basis of the distance, that minimum distance should be there, but central point is the mean of these points. Now the line will be drawn and now it will calculate distance of each data point, and where the distance will be minimum, that thing will be regarded as the first principal component. And why this direction is considered, because towards this direction variation is maximum and not towards this direction. So first principal component will be my this direction. Now I have to compute the second principal component. So for second principal component, again from the central point only I will draw lines into different directions and I will check that from where the distance is minimum and the variance of these data point is maximum, where they are lying different from one another but lying closer to this line. So similarly, second principal component will also be identified. Now my another task after identifying this second principal component is to transpose my data in such a way that my first principal component will come towards my x-axis. So in this way I will rotate my data set which was previously present like this to show it like this. So in this case, principal component 1 will be in this direction and principal component is in the x-axis. So means I am converting my slanted principal components to a proper horizontal and vertical axis. This is the thing which is done, and I will not call them as weight or height, I will call them as principal components. Yeah, so in this case there were only 2 dimensions, but in original data set you might have multiple dimensions. That's why you go for principal components, so that you can capture huge variance which is present in the data points. So before moving to supervised learning, I just want to make sure that whether it is clear. Okay. Yeah, so now let's see what does supervised learning means. So in unsupervised learning, we only have the data and we have to identify the patterns, we have to cluster it into clusters. But in case of supervised learning, we have the labeled information in which the data is already labeled, and by looking at that labeled data set, we will train our model. And now whenever a test data set will come which will give me a new input, I will predict that what could be its label. So that is about the supervised learning, where the user has given the labels and it is supervised. It has to now predict. So it has both the training and validation data sets labeled. So when we say validation, so it is also known as the test data set which we try to test our model. That so I have a bigger data set which have all the labels. I will divide it into training set and test set, or training set or validation set. So my validation set will be a smaller set from that entire data set, and training set will be a larger set on which I am training my model. So after training my model, for example, here you can see it is a labeled data, and in this each shape has some label. For example, for each shape I have mentioned that this is a hexagon, this is a triangle, this is a square, and in my data there will be multiple inputs which might have similar shape again and again being repeated. In model training, I will try to associate that if this is the shape, this will be its label. For example, if 3 lines are lying next to each other and they are closed, then it will be a triangle. So similarly, model training will be done. Now I will have test data or validation data which will be a part of my data set only, for which label information is already known. So I will use my training model, I will predict what could be its shape. For example, it is able to predict to be square and triangle, then I will check it because I already know what will be its label. So I will check if it is correct, that means my model is accurate. But if it is not accurate, then I will again train my model in such a way that in the end my results are accurate. So I'm trying to increase my accuracy. In this case it was quite straightforward, but in real scenarios there might be not such exact labels. So in such scenarios it was a categorical data set. But what if you have to predict the expression, or you have to predict the abundance? So in such scenarios the values will be numerical. So in that case values might not be exact, but they will be in certain range. So accuracy won't be 100%, but we try to increase the accuracy above 90% so that our model is better. So this is about the test set and training set. Now our model is trained, we have checked its accuracy, so now we are sure that our model is predicting better. So now we will have some new data set or some new input set for which we do not know labels. So now we will predict labels for that, and our model will be better and it will be able to predict those unlabeled data. So this is all about supervised learning, and it is divided into 2 categories. One is regression and another is classification, depending on what kind of data you have. So this is a table which contains the information of both the regression and classification methods or algorithms. So when we say linear regression, it tries to predict a continuous values. So when we say continuous values, which means they will be in the numerical form, in form of numbers such as protein expression. So such things are not like categories that high low, they are like numbers. This is the number of, or when we say mass to charge ratios, so there will be some numbers, there will be continuous values which will have some range, for example ranging from this to this. So for such scenarios we will use linear regression. It is simple, interpretable and fast, and it assumes linear relationships, and it is sensitive to outliers. So if you have any outlier, then also it won't work fine. Then comes logistic regression. So in logistic regression, it will be a binary classification, and it will be having the categorical output. It can either tell you protein is present or absent. It won't give you any numerical value. So for that we use logistic regression, and it is good for the binary outcomes. So now if in your data you only have to identify whether protein is present or not, then you can go for logistic regression. But if the output is not binary, for example if you are looking for the effect of a variation, it could be moderate, it could be low, it could be high, it could be extreme. So if there are multiple categories for which the output could be multiple categories, then you cannot use logistic regression. You can only use if there are only two possible categories which will be your output. So it is only good for the binary outcomes, and it is interpretable, and it also assumes linear boundaries, so it is not suitable for complex relationships. Because if the relationship is not binary, then you cannot use it, because in binary relationship you already know that equation mx plus c. So if it do not follow that equation, so in such scenarios you cannot use linear regression or logistic regression. Then comes support vector machines. So they are generally for classification. So if you want to classify proteins, then you can go for support vector machines, and they are effective in high dimensional spaces, and they are also robust to outliers. So they are better than linear regression because they can handle high dimensional data. They can have multiple uh, classes of your data set, or you can say multiple clusters only. So it is classifying your data into different classes, and it can be computationally intensive and harder to interpret in some scenarios. Then comes decision trees. So it is also a classification and a regression system. For example, if you are trying to predict the post translational modifications, so in the later slides we will see one by one that what does these algorithms are doing by taking examples. Then comes random forest. So when multiple decision trees are clubbed together and their average is taken for making some decision, then it is known as random forest. It is better than decision trees because I am taking the average. So it reduces the overfitting, and it handles large data sets as well. Then comes k nearest neighbors. So in k nearest neighbors also I will classify my new input set on the basis of its distance to all the available classes which are present. So if minimum distance is present to one of the class, I will call it as the, in that I will put it into that particular class depending on the distance, means what are its close by neighbors. So it is also simple, and no training phase is required because I'm simply looking at the its neighbors or the distance. Then comes naive Bayes, which is a text classification and functional annotation prediction. It is also simple, and it is based on Bayes algorithm. So Bayes probability equation which we have, so it is based on that. Then comes neural networks. They are generally used for the complex tasks, such as we have seen in the initial slides, that how neural networks were designed, how they behave like a human neuron. They can carry the information from one layer to another layer, and while doing so it tries to learn different things. So we will go through them one by one. And it is capable of learning complex patterns. It is highly flexible, but it requires large data sets, and it is also prone to overfitting. So now we will see linear regression, but before that, do you have any doubts? Okay, so now let's see what does linear regression means. So in linear regression you are trying to make a line from where the distance of all the, means you are trying to associate that how the how the values are related to one another. So in linear regression you have one dependent variable and one independent variable, and you are trying to find out what is the relationship of that dependent variable with the independent variable. So in this case I will make some equation to design a line which have minimum distance from these data points. So this line will decide whenever a new input will come. For example, in this scenario new input is here, so I will try to figure out what would be its output. So my x-axis represents my input and my y-axis represents output. So now how my x-axis is related to y-axis, and my x-axis will be my independent variable which is not depending on anything, and my y-axis will be my dependent variable which I am believing that it is dependent on my x variable. Now I am trying to predict my output based on the input. So now this straight line will be the line from where the distance of all these points is minimum. So it is assuming that it is a linear model, it is a linear distribution. So now this line is plotted. Now I have a new input here, so I will see that I will predict what could be its output. So I will try to extend it towards that line and I will predict this point to be my output. So this is the regression function. It computes the linear relationship between the dependent and one or more independent features by fitting a linear equation to observed data. Now in this scenario I'm trying to fit a linear line, but what if my data point is lying here? Then I won't be able to get this line because the distance of that data point from this line will be huge, and what my aim is to minimize the distance from other data points. So in such case I might shift my line to here. So it is sensitive to outliers. My outliers will affect my line because in the end there is a equation. So y is equals to mx plus c, where where m is the slope and x is my this independent variable and c will be my intercept. So by looking at that equation we believe that there is a linear relationship, so outliers will influence this equation. This line will be shifted, so it won't be able to predict exactly so what will be my output. So this is a linear regression in which I am predicting some numbers or some continuous variables, and I am taking the example if I want to predict the protein abundance. So suppose we have a data of mass spectrometry intensity values for different peptides which correspond to proteins, and we want to predict that what will be the overall abundance of these proteins in the sample. So now what it will do is it will use the linear regression to model the relationship between the intensity values of peptides and its abundance, because somehow we know that if intensity is high it means that more and more peptides are present, that's why its intensity is high. So in that way intensity can be related to the abundance. For example, in my data set, if for other data values if this is the intensity, so it means that these many number of peptides are present, so I can associate intensity with my number of peptides or abundance. So in such case intensity will become my x-axis or independent variable, and my abundance will become y-axis, which will be my dependent variable. So now in input means when I'm trying to predict, so in my prediction input will be my intensity value and my output will be what will be the abundance. So these all such kind of things can be done with the linear regression, where we are trying to predict some numerical value. And this is a table which tells you each detail that what will be input data, and I'm taking this in terms of the example from the mass spectrometry data, so you can go through this if you want to understand it in more detail. And I've also listed the equation, that how these equations means how actually it is associated. So I won't go into the detail, it is not that much important, but if you have any interest, so you can go through it. Then comes logistic regression. So it is trying to identify or classify your data into two categories or binary categories, which could be 0, 1, yes, no, present, absent. So this is just done for the binary classification, where we are trying to predict. So in terms of proteomics, it can be used to predict the protein presence or absence. For example, I have a healthy person and I have a cancer patient, so I'm just trying to check that which proteins are present in healthy person which are absent in the cancer person, or vice versa. So how it works, it could be applied to predict the probability that a given protein is present in the sample, and output will be a probability value, which is then threshold to decide whether the protein is classified as present or absent. So for example, we have the probability of protein which is present and the peptide abundance. So I will have both the values. In this case, I will be having both the x-axis and y-axis, and I will be joining these points, and I will decide at what threshold I will call it as a healthy or cancer. So this particular threshold is decided by us. So in this case, the probability threshold is 0.5. So if values exceed 0.5, which means that the probability of protein present is high, and which means protein is present. Whereas if value is less than 0.5, so we will call it as no. So these two categories are decided by us on the basis of threshold point. So in this case, we have taken it as a probability. We have taken a probability to decide that when we will call a protein to be present, and when we will call protein to be absent. So on the basis of data points which are already available, we will see the values and we will take their probabilities, and we will decide that when to categorize it into either of the category. So this is about the logistic regression, where we are dividing the data set into two categories. Is it clear? Okay. Yeah, so now let's so this is also a table which is giving detail about the logistic regression. If you are interested, you can go through it. Now we will talk about support vector machines. So in linear regression, what we have seen is that we draw a single line through which we are trying to kind of predict that what will be the output of the unknown data point. So in that case, we were having a single straight line, but in many cases a single straight line won't be able to capture the outputs. So in the support vector machine, we are trying to divide the data into different classes by taking multiple such lines which divides my data. So for example, in terms of proteomics, if we want to classify the types of proteins, so we have a set of proteins which are classified into different types, such as enzymes, structural proteins. So these are some classes, and I want to figure out that what will be my class of protein. So I will be having information of both the x and y axis. And now what I will do, so in these figures they have taken x1 and x2, so you don't have to confuse with it. These are just notations to refer to as x and y axis. So we already have the values, so these values will simply be the features. For simplicity, they are giving it in form of plots. So there will be multiple features which are allowing them to plot the data. So in this, for simplicity, they have given x and y axis. Now I have the already known features which helps me to identify that when to classify my protein as enzyme and when to classify as structural proteins, because I already have all the values with me. I already have the labels. I already know that these particular proteins are enzymes and these particular proteins are structural proteins. What the model is doing, it is trying to identify the pattern, that what are the features which are deciding that this is the enzyme, what are the features which are different among both these classes, so that whenever a new protein is given with features, I will be able to categorize, I will be able to identify the class. So this is the scenario. I already have all the information. What SVM will do, it will be trained on the labeled data to learn the decision boundary, that whenever all these features are present, then I can call it as class 3. When these features are present, then I will call as class 2. Similarly for class 1. So we will check for all those features which are deciding features for a particular class. So these are the support vectors, which are support vectors will be the points which are present in each class. They are known as support vectors. There will be optimal hyperplane. Optimal hyperplane is a point which is trying to segregate or distinguish your two different classes. And then there will be a distance. among the optimal hyperplane and your support vector. So these support vectors will be the terminal points of your class or a cluster, which are lying at the ends, because these are kind of support vectors. If any point which is present closer to this line, then it cannot be classified into a class. That will create some issues. So this is the maximum margin which should be present from the hyperplane. This should be the maximum margin, so that you can identify or you can classify easily. But if data points are lying in the hyperplane, then you won't be able to decide that to which class you can put it. So in such case, there will be some issues with the identification of hyperplanes, because hyperplanes should be such lines which could help you to distinguish among two classes, and there could be multiple classes also. So in SVM, it can also be used to for the multi-class classification. So that's why there could be more than 3 classes also which could be present, and your aim is to identify the hyperplane which could help you to distinguish among different classes. And this hyperplane will be based on the features which is already available with you. So you will only consider those features which will distinguish among these classes. So in some case, all these techniques are kind of integrated together. So you can firstly use PCA and then use SVM. Firstly you can reduce the dimensions, then you can go for SVM for classification. So these techniques can be used in conjunction to one another. And this is a table about SVM if you want to go to the detail. Then comes decision trees. So these are like a tree kind of architecture in which on the basis of the input which is available, you try to identify the output, that what could be the output. You will make a decision. So their structure will be a flowchart like structure which are used to make the decisions or predictions. So for example, I have this data in which I have gene pairs. I am looking for their interaction, that whether they interact or not, what is their expression correlation, what is whether they share some location where they are present, or whether they share functions, and what is the genomic distance among them. So now what I can do with this available data, I can construct a decision tree. For example, if my expression correlation is greater than 0.9, so then I can make a decision that yes or no. So when expression is greater than 0.9, if yes, then I can look for the shared function, that whether they share some function or not. So similarly, there could be two outcomes which could be yes or no. So for example, if we take this example, then you can see that in the case of in second point, the expression is greater than 0.9. Now I am looking for the shared function. So this is yes, and yes for AC. Similarly, it is no for both CD and AB, but their expression is less than 0.9. So in this no, we do not have any point or any value, any gene pair. Similarly, if expression is less than if it is not greater than 0.9, which means AB and CD, then what I will look for, I will look for shared cellular localization. So it is based on you, that which feature you are observing. So in this case, they are observing shared cellular localization. So in case of AB, yes, there is shared cellular localization, whereas in case of CD, there is no shared localization. So no CD will come. In yes, we will look for the genomic distance less than 5 kb. Okay, so in case of AB, yeah, its distance is less than 5 kb, it is 1 kb. So in yes, AB will be captured. So now what it is doing is, it is trying to identify the decision for these gene pairs. So in this case, we only have 3 gene pairs, but in reality you will have multiple gene pairs or multiple input sets, input data, for which you will design a decision tree. Now if you will get any new value or any new gene pair, then you will be able to predict that if their expression is greater than 0.9, then you will be able to predict whether they will share a function or not, on the basis of data which you will be having. So if yes count will be high, then in the next time whenever you will find the expression correlation greater than 0.9, you will directly say that yes, the probability of them sharing the similar function will be high. So this is about the decision trees, which helps you to make decisions or predictions on the basis of already available data. And their example is the determining protein post translational modifications. For example, if you have the features information from mass spectrometry data which tells you about potential post translational modifications on proteins, and you want to classify which types of PTM is present, then you can design the decision tree which will split the data into different features such as specific mass to charge values and retention times, and they will determine the type of PTMs on the protein. So on the basis of what is their mass to charge and what is the retention time, they will decide that what kind of PTM will happen. So in the next case, whenever you will give some input, so the decision can be made on the basis of that already designed decision tree. Now what can happen is, if you have a single sample data set, then it is quite easy to make a decision tree. But what if you have multiple sample information and you have multiple data sets, and in some cases it go with yes, in some cases it go with no, then how will you decide? So in such cases we go for random forest when we have multiple training data sets. So we will have multiple decision trees and we will try to average them. So what will be the average will become my prediction. So this is random forest. When we have a huge amount of data, then we can go for random forest, and when we have a single data, then we can simply use the decision trees. And in this case, for example, we can predict the protein-protein interactions. So this is about the random forest, and we won't waste much time here. Then comes k nearest neighbors. So simply in this also we will simply look for the distance. So it tries to identify the patterns, and it tries to identify the similarity between the new data for which you want to identify, for which you want to classify it into already available classes. So you will find the similarity of that new point and the available cases which are present, such that you can put this new case into the category which is most similar to the available category. So in this blue dot represents the new data point, and you have category A and B. You will calculate its distance from the both these categories. So the points which are nearest to this from both category B and A, we will find their distance. So now in this case, the minimum or the nearest point from category B will be this point, and from category A this point. Now when we will find the distance, so distance from this point is minimum as compared to distance from category B point. So we can classify it into category A. So in this case, means in this figure it is quite difficult to interpret the distance, but in reality you will be able to identify that distance on the basis of features. So k nearest neighbor means you are trying to identify its nearest neighbor, and to which category it will be nearest, it will be classified into that particular category. So this is about k nearest neighbors. Then come naive Bayes. So it is based on the Bayes theorem, which is kind of a probabilistic classifier which gives you the probability of an event given that there is some prior probability or prior information of certain event which already has happened. So you already have that information and you are trying to predict the probability of the new event on the basis of already happened event. So that is about the Bayes theorem, means the concept of Bayes theorem. So in this naive Bayes, you try to so this assumes that the occurrence of certain features is independent of occurrence of other features. So in case of proteomics, you can use it to predict the functional annotation of proteins. For example, if we want to predict that whether my protein is an enzyme or whether it is a transporter, so I can go for naive Bayes. I have certain information already available with me, so I will calculate the probability of each functional category given the observed features. So on the basis of features, I already know that these features correspond to this category. So now with the already available information of those features, I will be able to predict the probability, that what is the probability if given these features my given protein will be an enzyme or a transporter. So it is a probabilistic classifier, you can say. And for quick detail, I will give you the example. So this is the example of a person when this is the weather, that what is the weather, this is the temperature, this is the humidity and the condition of wind. So this is the outcome or the decision or you can say these are the categories, yes or no outcomes of a person, that whether he will play golf or not, given that these are the weather condition, these are the temperatures, these are the humidity, and these are the winds. So in case of naive Bayes, we design the likelihood tables. We have frequency table with us and we design likelihood table. Likelihood table gives you the probability, that what is the probability if the weather is sunny and person will play. So yes or no, for which it will give you the probability value. And for detailed understanding you can go through these slides. In this each and every point is mentioned in detail. So just I will explain the topmost table, that how it is calculated. So what we do is, one by one we take each column. So firstly we are taking the outlook. So outlook is nothing but the weather. What is the weather condition? So sunny, overcast, or rainy. So in this firstly we will see what are the unique labels which are present in terms of outlook. So unique labels were rainy, overcast, and sunny. So there are only 3 unique labels which are present in this column. So we will construct a frequency table by taking 3 unique labels. Now what we will see? We will see the frequency of playing golf, yes or no. There are 2 possible outcomes, one is yes, one is no. Now we will see how many times when the outlook is sunny, then the person is playing golf. So for sunny the number of events are 5. Okay, they are 5. And how many times he is playing in sunny? Yes, 2 and then 3. Yeah, so out of 5 sunny days he is playing 3 times, that means 2 times he is not playing. So 3 and 2 will be captured in this way. Similarly for overcast also we will see that how many times he is playing. So 4 times he is playing and 0 for no. Similarly for rainy also we will see. So rainy is also present 5 times, out of which 2 times he is not playing, 3 times he is not playing, and 2 times he is playing. So similarly frequency table will be created. Now how I will get the likelihood table? So for likelihood table, firstly we will see that how many so how many times yes is coming. So total yes probability of person playing is 3 plus 4 7 plus 2 9. So 9 times he is playing golf, 5 times he is not playing golf. There are total 14 events which are present. So out of these 9 events, now this particular likelihood table is giving you the probability that the person will play when the person is playing, given that weather is how many times he is playing, given that he is playing, and what is the probability that the weather is sunny. So in this way probability of yes will be my prior probability which is already known, and probability of sunny means how many times he is playing that is sunny. So 3 will be divided by 9. So if you have seen the lectures of Bayes theorem, then you will be more clearer. So you can consider those lectures, and I will try to give you the video link so that it will be more clear, because it is about the statistics, so I can't explain the statistics in this class. Yeah, so similarly we means in this what we do is we have a prior probability, the event for which we already have information, and we try to predict the probability of the next event. So in this scenario, for all these cases, they are saying that given that person is playing golf, what is the probability that these many times he is playing and the weather will be sunny. So that thing is being calculated in the likelihood tables. Similarly in the next slides you will see that for each kind of or for each column the frequency table is created and likelihood table is created, and ultimately all these tables are combined together, all the likelihood are combined together. So what is the probability that he will play, given that all the cases, all the conditions, any of the condition could be there. So what is the probability he will play and what is the probability he will not play. So these means these decisions or these probabilities are calculated in naive Bayes. And in this case another thing which I want to mention is that they are multiplying the probabilities, which means that these events, they are independent of one another and they are not mutually exclusive. In case of mutually exclusive events, we kind of add the add different probabilities together, whereas in case of independent events we multiply the probability values. So that is one assumption of naive Bayes, that whenever it is predicting the probability, it assumes that all the events which are taking place, they are independent of one another. So this is about naive Bayes, and don't worry about all these slides because this is a very detailed slides, but you won't be asked for all these details. This is just for your understanding that what is done in naive Bayes. Then comes neural networks. So as we have discussed, that neural networks kind of replicate similar structure as that of our human neurons. They also have the dendrites. Dendrites are in, if we talk about brain, then dendrites are the initial points of the neuron which capture some signals. So dendrites receive signals, then they transmit it through their cell body through axons, and then from these synapse another neuron dendrite will be connected, so information will be passed on from one dendrite to another dendrite of another neuron. Similarly in neural networks also, the dendrites will be my inputs, means the signals which dendrite are capturing will be my input points. They will have certain values, and my function will be my body of the neuron, and then there will be some output. So these are the concept of neural networks, that there are certain inputs, and on the basis of those inputs I will decide that what will be my output. So output will be a single value, and for this I can give you a small example. That for example, if I talk about identifying the patterns, for example you have an image in which there is a 8 being drawn, and you want to identify what is the digit in the figure. So in such case the inputs will be the dots. So what you do in case of such pattern recognition, you divide your picture into different pixels or different small cells, and you will see which cells are darker, and you already have the information of cells which are darker for each digit. Now what you will do for this, all these inputs will be my points where the cell is darker. So I will simply check these inputs, I will match them to my already available data set, and to my already available data set I have some functions. So my output will be a single digit. So input could be multiple, but my output is only single. So that is the concept of neural networks, that you give multiple inputs, but in the end you just get a single output. And there are different types of neural networks, I won't go into the detail. So in neural networks also you have the linear regression models and other statistical equations also you can use depending on that what is your output. But in this case you can see that multiple inputs could be there, but output will only be a single value which will tell you. For example, if you have multiple sequences and it is telling you that whether the protein is expressed or not, so value output will be yes or no, or it will be some number. So some single output will only be there in this case. So example is protein sequence classification. For example, different sequences are given and you want to identify which protein is there or what is the name of the protein, so output is only one. So this is about neural networks. And one more thing which I want to add is that all these means inputs are known as nodes. So these are known as nodes which are connected, and this is a paper if you want to understand that in the latest research how these neural networks are used. So this is one of the paper in which they have mentioned about biologically informed neural networks. So this paper was published in Nature Communications, in which they are trying to interpret biologically informed neural networks for enhanced proteomic biomarker discovery and pathway analysis. So they are trying to identify certain biomarkers for which they are using the neural networks. So in neural network also you can classify and you can also predict the values. So different things you can do with the neural networks. So first step which they have followed is the data collection and reanalysis. So in this case they have taken the data from patients who have septic AKI and COVID-19, and then they have reanalyzed it and converted into the data set for each disease. Then comes bin generation. So bin is the biologically informed neural network. It is created for each data set by selecting the relevant pathways. So there could be different features which are associated with these diseases. So in this case they have taken the pathways.
that which pathways are influenced in AKI versus COVID-19, so that they can distinguish between both these diseases. So similarly, they have looked into the pathways, and they have structuring them into sequential neural network models that reflect the proteomic content of the dataset. Now they have trained this neural network where the protein quantities from each sample are used to train bin to distinguish between two subphenotypes of the disease. And then for interpretation and analysis, the trained networks are interpreted using the SHAP, which is Shapley additive explanations, to determine feature importance. So you don't have to go into such technicality of the terms, but overall what they are doing is that they are trying to distinguish among two subtypes of the disease. So one is the AKI and one is the COVID-19, and they are looking for the features which are altering in AKI and the features which are altering in COVID-19, so that they can distinguish among both of them. So with the help of neural networks, they are trying to identify that how these features are related to both of them, and how they can predict by having a new value, how they can predict whether it will be a part of AKI or whether it will fall into COVID-19. So in this case, they have used the neural networks to classify it. And this is a concept booster map in which you can identify that what means method you can use. So for example, if you have labeled data, then you can go for supervised learning. If you do not have labeled data, then you have to use unsupervised learning. Similarly, when you are talking about supervised learning, then you will see that what will be your output or what kind of data you are analyzing. So if it is a categorical data, which will be a categorical value, such as it will have some classification or some yes no kind of thing. So in that case, you can go for classification. Whereas in quantity, if you have a quantity, you want to predict some numbers, then you will go for regression, and these are certain methods under regression. Then comes unsupervised learning. So if you want to group your data, then you will go for clustering. If you want to reduce the dimensions, you will go for dimensionality reduction. So this is about the learning methods, different types of learning methods. And now we will see that means what should be your machine learning model, because you are kind of designing models by using these methods. So there are chances that you can go for underfitting, where you're not able to properly classify it or properly cluster it, such as in k-mean clustering, in linear regression. So these are certain methods where you are kind of so stringent, you are assuming that your data is linearly distributed, but it is not the case. So in such scenarios, you miss on multiple data points. So when you miss multiple data points, that is known as underfitting of the model, when you are not able to capture maximum data points. Then comes a good model in which you are maximally able to classify it, or maximally able to means you are properly able to distinguish among two classes, or you are properly able to get the output value as in case of linear regression. So that is known as a good model. Whereas there are chances that you can overfit your model, for example, as we have seen in the Gaussian mixture models. So if there are outliers, then it will try to even take that outlier also into the cluster shape. So in that case, even if you are taking the data point, but it is not of any relevance, and it is also disturbing the shape of your cluster. So in certain cases, noise can also come in. So that is known as overfitting. So even if loss is low, but accuracy is also low, so it is not capturing the variation in a proper way. So these are three kind of conditions which can happen during machine learning models. So now what we have to do is, we have to go for hypothesis testing. So whenever we use any of these classifiers and any of these models, then we also have to be sure that whether my prediction is accurate or whether it is not accurate. Because in research, our main aim is to design a new hypothesis, and then there is a two kind of hypothesis. One is null hypothesis and one is alternate hypothesis. So alternate hypothesis is a modern hypothesis which you think of, and null hypothesis which is already prevailing. So you have to reject that null hypothesis. You should be so sure that that null hypothesis should be wrong. You should be able to disapprove that, so that your alternate hypothesis could be accepted. So whenever you are using these models, so you have to be sure that whether your prediction is right or not. So in such cases, you have to go for testing to make sure that the results which are coming, they are statistically significant or not. So in cases where they are not significant, you cannot go with those examples, and your hypothesis, alternate hypothesis, will fail. It can't be accepted, so you will lose. So hypothesis testing is a statistical method which are used to make the inferences about a population based on sample data. It helps you to determine that whether there is enough evidence to reject a null hypothesis in favor of an alternate hypothesis. So these are the steps that what is done in hypothesis testing. So first step is formulate the hypothesis. So there are two kind of hypotheses which I mentioned. One is H naught, which is the null hypothesis, which asserts that there is no effect or difference. So this is given in terms of example, that for example you are saying that the normal person and the cancer person, they have variable protein expression, but null hypothesis say that there is no variable expression. This is just for your reference, just an example. So null hypothesis asserts that there is no effect or difference, and alternative hypothesis is that there is an effect or difference. So you are trying to prove that there is a difference. Now you have to choose a significance level or a threshold, so that if that particular threshold is not met, so you cannot accept your alternate hypothesis. So there is some significance level which must be satisfied, and it is normally set at 0.05. This is the threshold for deciding whether to reject H naught or not reject. So in the later slide I will show you that means where the alpha lies on the basis of the graph. So next case is to select the appropriate test. Depending on what you are trying to analyze, what kind of difference you want to see, you can select any one of these tests. So first is t-test, when you want to compare among two groups and compare their means. And in this case, if you have the parametric distribution, or when the normal distribution is available, this is for the parametric tests. Then comes ANOVA, which is for comparing mean across more than two groups. So then you can go for ANOVA. When you have a categorical data and you want to assess the associations, then you go for chi-square test. Whereas Mann-Whitney test is non-parametric test. When your distribution is not normal, then you can go for Mann-Whitney U test to compare among two groups that what are the differences, whether there is difference or not any difference. Now the fourth thing which you do is you calculate the test statistics, where you measure the degree of agreement between the sample data and H naught. So in this you try to figure out that whether your H naught and H1, which so your test is corresponding to which of these hypotheses. You determine the p-value. So p-value indicates the probability of observing the data or something more extreme if H naught is true. So low p-value, then you can reject H naught, and if your p-value is high, then you cannot reject H naught. So p-value, to some extent, in simplicity, p-value is like a probability which says that this kind of distribution might come by random. So if something can occur random, so you cannot be confident about it. So your p-value should always be less, and in journal its threshold is taken as 0.05, but it depends on you, because whenever you are making some hypothesis, you have to make sure that the result should be better. So in that way you can alter these values. In some cases you can also go for the strict p-values, which could be less than even 0.05. So means you're trying to improve your hypothesis, so for that you can select these p-values. Then you have to make a decision. You have to compare the p-value to alpha. So alpha is the significance level which you have decided earlier. If p-value is less than or equal to the alpha, then you can reject H naught. But if your p-value is high, then you cannot reject H naught, which is the null hypothesis. And in the end you have to interpret your results, through which you can decide that whether your hypothesis is right or not, whether you can reject null hypothesis or not. Now we will look into the errors and that what kind of errors you can see. So for example, this is the distribution. In this case I am assuming that my data is distributed normally, so these distributions are showing the normal distribution. So this is the null hypothesis and this is the alternate hypothesis. Now what can happen is that in certain scenarios my both of them could overlap on one another, and there is a critical value, so above which, if critical value is kind of alpha, which we decide that this is the significance value. If anything will lie above this alpha, then I will accept my alternate hypothesis and I will reject null hypothesis. But if value is below critical value, then I cannot reject null hypothesis. But what can happen is that even if the value which is coming, if it is below the critical value, if your point is below the critical value, then also there are chances that it can satisfy alternate hypothesis. But still you have decided your critical value to be here, so in this case also you will not reject null hypothesis and you will accept null hypothesis, whereas there are chances that your alternate hypothesis could be correct, so which can lead to type 2 error. So type 2 errors are also known as the false negatives, which means you are terming it as negative but it is not negative. So these false negatives, these negative, true negative, false negative, they are talked in terms of alternate hypothesis. So false negative means when your the decision about null hypothesis, you do not reject it but null hypothesis is false, so it is a false negative. Now when I say false negative, I talk it in terms of alternate hypothesis, which means that I am rejecting it, but it is not negative, it is false negative, and this will lead to my type 2 error. Similarly, if anything will lie at this point, which is from here to here above the critical value, but still it can means come under null hypothesis, so this can lead to type 1 error, where I am rejecting my null hypothesis but there are chances that null hypothesis could be correct, so this can lead to type 1 error, and this is also known as the false positives. So when I say false positive, that means I am accepting my alternate hypothesis, which means I am saying it as positive but it is false because null hypothesis could be correct. So this is known as false positive and it is known as type 1 error. And what is the probability? Its probability is alpha, and then comes true positive. So true positive means when your null hypothesis is also false and you are also rejecting it, so which means true positive, that your alternate hypothesis is positive, that means you have accepted it and it is also true. So this region where the correct decision can be easily taken above the critical value is your true positive, whereas the values below this, they could have some consequences, they could be either they could come under type 1 error or type 2 error. So these are the chances, that how many means there is a probability that null hypothesis might be correct. It is not sure that it is correct, but there are chances, so that's why these are known as type 1 and type 2 errors. And to reduce these errors, we have certain corrections which we have seen last time, such as Bonferroni corrections, Benjamini. So such corrections come into play, which tries to change your p value as well as the critical value in such an extent so that you can avoid these errors. So this is a figure in which it is mentioned in detail, so you can go through this figure, it will make you more clear that what each of these points mean. Okay, now I will talk about ProTIGY. So till now, is everything clear? When you will go through these slides, then you will be more clearer, because in this class you might not get everything, but when you will go through your slides at home, then it will make you more clear about the concepts. Now let's see about ProTIGY. So what is ProTIGY? It is a software tool which is generally used in mass spectrometry for accurate protein inference and its quantification. And ProTIGY can accept these file formats, which could be a text file or GCT file, and in text file it can take TSV, CSV, TXT, SSV. So these are certain formats which are supported by ProTIGY. It analyzes the, so if you have any quantification or means mass spectrometry data, you can simply provide it to ProTIGY and it will help you to automate the identification of proteins and quantify them. You can also look for their differential expressions. So for all such analysis you can use the ProTIGY. And now in the later slides I have shown that how you can, what advantages ProTIGY provides you and how you can go through it. So through this link you can go through their GitHub page, and in the GitHub page only, if you click on the readme, then you can read all the instructions which are available. And in their projects only, they are also giving you the entire PDF, that how the ProTIGY works. So in this I do not have any example of mass spec data, so because it also require your account in the broad site. So if you do not have any account, you cannot use it. So in this we are just understanding that what ProTIGY provides you, what advantage it provides you, but if you want to use it, you can follow the instructions which are provided by them in the manual. So this is the main site of the ProTIGY, and right now it might have changed because I do not have account, but these slides are provided by them, and it might have, means till now this version might have updated, so this home page might vary, but previously it was like this. So you can upload your file, which could be in any one of these formats, and you can save the session name and you can import it, then you can go for its analysis. So according to them, the first step is that you must have your broad ID. You can simply log into it. You should have the R in your system, and then you can export your results into either HTML, PDF or Excel data formats, because you have multiple samples and their mass spectra. So after the analysis it can provide you the differential expression. So all those things can be in the form of tables, which could be in the CSV format or in the Excel format, or you can also export it in the PDF format. So that are the results, that how you can export them. And what kind of aspects it cover about data analysis: it can tell you about quality control, it can also transform or normalize your data which is present, it can filter the data, and different test statistics you can perform, and you can also visualize your data, such as you can go for the volcano plots or PCA. So these are the stepwise workflow of the ProTIGY. It imports the data, it decides experimental design, so you have to select all those things. There are some filters from where you can select. It can go for gene mapping from the RefSeq or UniProt. You can also select the organisms for which you have provided the data. It will manipulate the data, it can perform log transformation, normalization, filtering. So you can select any one of these techniques for manipulation. You can select the markers if you want. You can also decide the significance filter. You can decide what should be the p value, what should be FDR, what should be the top n, so what should be the top number of fields you want to see. You can perform exploratory data analysis, you can go for quality control, and you can export your data in the end. So there are certain things. So when you will be working with ProTIGY, you can select any one of them based on your desire, that what analysis you want to perform. And yeah, this is the home page where you can select from these categories that what you want to do with your data set, and then you can simply run the analysis, and in the progress bar you can see that how much progress has been achieved and when this progress will be done. Because if you have a smaller size of data set, then the progress will be faster, whereas if you have a larger data set, then it might take some time. Then simply you can navigate through these tabs which are present, depending on means what you want to see, whether you want to visualize or you want to cluster the data which you have provided. So all those things can be done in ProTIGY. You can also set the significance thresholds, as we have discussed, the p values, and then the analyzed data set will be shown in these terms, such as number of features, number of expression columns, number of groups, and it will also tell you that what kind of normalization it has performed, what is the number of significant DMSO drug. So these are some data sets, and then it can quantify the number of data points and the data columns, so different drugs and what is their expression. So all those things can be exported into PDF or CSV format. So this will be the output which it will provide you after progressing. And this is just to show you that you can also go for volcano plot, which is across the log p value and the fold change. So you can see that which proteins are overexpressed, which are underexpressed. So all those things can be done and observed. So this is all about the ProTIGY. Now we will practice few MCQs and then we will end the session. So till now, is everything clear? Okay, thank you. Yeah, so now I will wrap up. So first question is, which of the following is incorrect in regards to machine learning algorithm? So option A is class discovery is linked with unsupervised learning, whereas class prediction is linked with supervised learning. Option B is unsupervised machine learning needs a training dataset to learn and predict. Option C is k-means clustering is an example of unsupervised learning. Option D is regression and classification comes under supervised learning. So what do you think, which among these options is incorrect? I think this MCQ will also help you with your assignment, because somehow a linked question is there in the assignment. So if you are able to answer this, then you can answer that question also. Okay, then now let's see what is the correct answer. So correct answer is that unsupervised machine learning needs a training dataset to learn and predict. This is incorrect, because in unsupervised learning we do not need a training dataset because we do not have labels. So this is done for unlabeled dataset. So in this we do not need a training dataset to learn and predict. There is no prediction. It is trying to cluster your data. So this is a prediction method, means kind of a it's kind of a class discovery method, not a prediction method. It's a class discovery method in which you have a data set and it will try to cluster it into different classes, and it will discover those classes, that which are the points specific to a particular class, so that if a new input is provided, it can classify it into any one of those classes. Whereas in terms of supervised learning, we already have a labeled data set, so we have a training set and test set, and we predict the new value which is coming, so we predict what should be the output. So that comes under supervised learning. And yes, k-mean clustering is an example of unsupervised learning. And do not confuse the k-mean clustering with the k nearest neighbors. So k nearest neighbor comes under supervised learning, whereas k-mean clustering comes under unsupervised learning. So the clustering and the dimensionality reduction, they comes under unsupervised learning, whereas regression and classification, they comes under supervised learning. Okay, so now let's look at the second question. Which of the following is false regarding hypothesis testing? Option A is a successful test always reject null hypothesis. Hypothesis testing always start using the population parameter. Option C is alternative hypotheses don't play any role in hypothesis testing. And option D is derive the distribution of the test statistic under the null hypothesis from the assumptions. So you have to tell which of the following is false regarding the hypothesis testing. I think this will also help you with the assignment, because some part is mentioned in this. Okay, so alternative hypothesis don't, yeah, right, because during hypothesis testing both of my null and alternate hypothesis, they play an important role while testing it. So yeah, this one is false, and rest everything is correct. Yeah, so now next question is that there is a decoy database, which is a database which looks similar to the target database but only contain peptides which cannot be the part of the sample. Given below is a target database of proteins. Now which of the following could be corresponding peptide decoy database? And these are the two peptide sequences which they have given, and you have to tell that which among them could be the decoy, or either they both could be the decoy. Okay, so I won't take much time. So its correct answer is both 1 and 2. Now I will tell you that what is a decoy database or what is a decoy sequence. So decoy sequence has a similar composition to that of the sample sequence, but what we do is we alternate the pattern of sequence. We alternate the way in which those sequences are arranged, such that we are trying to check that whether the peptides which are fragmented by mass spectrometry, whether these are randomly generated or either these are accurate. So for that reasons we should have a decoy sequences available in our sample, so that whenever we are going for the mapping of the peptides mass to charge ratio to their sequences, so we should be sure that that assignment or mapping is accurate or not, because if they are mapping to decoy sequences also with higher amount, so that means I am not able to properly map my peptide fragments generated by mass spectrometry. So that's why decoy database has important role to verify your results. So in this scenario, both of them, because if you look at the composition of their residues, so they will be similar to both. So in this scenario they are similar, and in journal the practice is that you simply reverse your sequence to design the decoy sequence. But otherwise you can also just means maintain the amino acid composition and just randomly rearrange the pattern of, or the rearrange the sequence or the series in which the sequence is arranged. So both of them will act as a decoy sequence. Now the next or the last question is, Ankit wants to perform the proteomics analysis of breast cancer data, and it is deposit in repositories, and which among of the following can be used to download the proteomics data: PRIDE, Proteome Exchange, TCGA and JPOST. So now you have to tell that which of the following repositories he can use to download the proteomics data. I think this was also in your assignment, I'm not sure. Okay, now let's see. The correct answer is A, B and D, because TCGA we already have seen that it is for the genomic data, not at the level of proteomics. Whereas PRIDE and JPOST, they are dedicated proteomic data repositories, while Proteome Exchange is like a consortium where it collects the mass spec data from multiple sources, and TCGA, as we know, it is for the genomics dataset. Now I have included the JPOST database because in your lectures I think we have not seen the JPOST, so this is for your reference. It is a Japan proteome standard database, and it contains reanalysis results with unified criteria for proteomic data. So it will also provide you the proteomic data, and peptide and peptide sharing among proteoforms will also be provided. So this is the home page of this data, and I have also given the link to this database in the bottom, which might be you can see that in the slides. Till now it's not visible. Okay, and there are some add-on concepts because I have seen in your assignment there were few questions which were like other than which were not covered in the week 5 topics. So I have seen the protein quantification and qualification application compendium given by the Thermoscientific, in which they have mentioned that for the by looking at the absorbance for the peptides and protein measurements, there are 3 different methods. One is E20531 method, one is Scopes method, and another is custom methods. So in this they have mentioned that in term of Scopes method, what is done is the measurement is taken at 2 different wavelengths. So one is 205 and another is 280, and they have also mentioned that what is the significance of taking the reading at 205 wavelength, because it is taken because at that wavelength there are some residues in the protein such as tryptophan, tyrosine, which are aromatic in nature, which have a benzene ring kind of a structure, so they only absorb at this wavelength. So whenever you are calculating the absorbance by using Scopes method, so you take the ratio of both absorbance at 280 and 205 ratio in the equation to correct for the tryptophan and tyrosine side chain absorbance. So I hope it might help you, and I have also provided the link. So if you want to go through this compendium, you can go through it to get more details. And ultimately I have attached that how proteoforms are identified from proteomes, and there are 2 type of methods, top down and bottom up. So bottom up stands when you try to identify canonical proteins when you already have proteoform information with you, whereas top down methods are the ones when you have the protein level information and you are trying to identify proteoforms. So these are 2 types of methods, and in this paper you can read it, it's a good review. I have attached its link. In this they have shown that what are the post translational modification that can take place, which can convert a protein into multiple proteoforms. Yeah, so this is all about today's session. Thank you so much. Do you have any doubts? Yeah, it was there in the assignment. I think we have also discussed in the initial lectures where we have mentioned that if you want to identify a novel peptide, and because in mass spectrometry the already available database is very important, to map the peptide spectra to a sequence. So when you do not have that protein, whenever a protein is novel, so in such scenarios what you can do is, first thing is that you look for the homologous sequences, and if homologous sequences are present, then you can use the homologous sequence information. And second thing is that you can go for the of translations, so theoretical translation of the genetic sequences. So you can go for 6 frame translation or 3 frame translation. So by going through the translation of the DNA sequences or the RNA sequences, you can design a theoretical protein sequence. So even if it is not experimentally present in some database, but then also you can predict that this, there is a possibility that this protein sequence could exist. So in such means in such cases where the database do not have that protein sequence, you can get a theoretical protein sequence. So which algorithms you are talking about? So you are talking about these machine learning algorithms or some other algorithms to do okay, machine learning algorithms. Yeah, so these are not kind of softwares. First of all, there are some codes. So in Python programming, or there are some means online available Python libraries which are already available, in which codes are written. So you simply have to provide your training data and test data into that. So these are command line things, not exactly software kind of things. So in command lines you decide that, yeah, so there are different functions where you decide that which statistical test you want to perform. So in even in neural network also, you can go for linear regression or some logistic regression. There are many other functions which you can decide, but these all things are done at the level of coding. In most cases they go for the Python programming. And when software is provided to you, then it means that model is already created. Then there is nothing to do in machine learning, because in machine learning you are trying to design a model, and software is like a already designed model. So yeah. Okay, thank you so much. So I will end the session and see you next week.